Today we are going to discuss the dental pulp. I'm Dr. Hibalter. I will be your host for this subject. As usual, we will start by discussing our outline. First, we will talk about the organization and composition of the tissue. As usual, I will introduce you to the building block of the dental pulp, which is, as any other connective tissue, a group of cells lying in an extracellular matrix. Then we will talk about the blood supply and the nerve supply of the dental pulp, the histology of the dental pulp under the microscope. There's different regions of the pulp we are going to see and discuss. And finally, we are going to talk about the age-related changes in the dental pulp. The dental pulp is a specialized connective tissue. It is contained within the tooth, under the enamel and the dentine, in the crown and in the root. Apically, it is continuous with the periodontal ligament you are going to study in a few weeks. This dental pulp is responsible of forming the dentine by a special kind of cells called odontoblasts that we will discuss in a few minutes. If we look at the periphery of the dental pulp, or the junction between the pulp and the dentine, we will find a layer of columnar cells, polarized cells, covering the whole periphery or line between the pulp and the dentine. These are the odontoblasts, which are responsible of forming the dentine. Moreover, and at the periphery, we can see two other components that we are going to discuss, which are the nerve terminals and the dendritic cells. Underneath the odontoblast, we will be looking at the connective tissue, the loose connective tissue of the dental pulp, also is going to be discussed. As you notice, and I think everyone knows, every one of you have actually extracted a tooth one day, or saw someone who is extracting a tooth. So we all know that at the end of the roots, we have a foramen. This foramen is called the apical foramen. It's where the nerve and the vessels enter the tooth and the branch inside the root and the crown. They both enter together in what is called the neurovascular bundle. But in addition to the apical foramen, there is accessory canals found on the sides of the root, and also sometimes there is some foramens in the area we call furcation that enters from the bone here that lies between the roots, especially in the posterior teeth. So what is the composition of the dental pulp? Actually, the dental pulp is composed from 75% of water and 25% of organic material, which are actually cells lying in extracellular matrix. You have studied extracellular matrix in cytology and general histology, and you all know that extracellular matrix is a versatile group of polysaccharides and proteins secreted by the cells of the tissue. This group of polysaccharides and proteins, they assemble a framework or a scaffold for the cells to sit and function and make the tissue, the architecture of the tissue. It's actually in control of the activity, the development, the migration, the division, and the shape and function of all the cells within it. In dental pulp, collagen is the predominant component. It's almost 1/4 to 3% of the dental pulp. And it has two components, the fibrous and the non-fibrous components. The fibrous components is mainly of collagen type 1 and collagen type 3. There is small amounts of type 5 and 6 and fibronectin and fibrillin. These details are discussed in the book, and they are more in the field of cytology. So I'm going to leave the details for you. You have to read them as homework, and I am going to ask about them in the exam. The non-fibrous extracellular matrix component is mainly composed of glycose, aminoglycans, protoglycans, and glycoproteins. Also, the details of these components are mandatory, so please go back and read them as a homework from the book. So the second component of the cells, there is four main cell components of the dental bulb. Of course, the hero of the dental bulb, which are the odontoblasts. Remember, they send their processes in the dentinal tubules in the dentine, the tissue that we have discussed last week. The most abundant cell of the dental bulb, which is the fibroblasts, some immune cells, and some populations of stem cells. We can see all of them here. So these are the odontoblasts. Here is the fibroblasts, immune cells like T cell, macrophages, dendritic cells and neutrophils, and some stem cells. So the odontoblasts, as you can see here, they are columnar polarized cells. Actually, they are fully differentiated cells, which means if they are insulted or injured, they die, they can divide. What does the polarization mean? It means that the plasma membrane actually have some invaginations which divide the cell into different compartments that have different functions. As you can see here, the cell is in the basal area of the cell, and the secretory organelles are at the top. So they perform their function in secreting dentine. See that the odontoblasts get some attachment to each other and to the fibroblast beneath by different kind of junctions like gap junctions, desmosomes, and tight junctions. When we take a look at the odontoblasts in a histological section, we see them as a multi-layered columnar cells, which are called pseudostratified. Why is it pseudo, which means not a real one? Because when we cut the section, it's impossible to cut a section in a thickness of one cell. So the thickness that we can do, it's like from 4 to 6 micrometers, which actually include multiple layers of cells above each other. That's why we see this pseudostratified seen under the histological in the histological section. So as you can see, these are columnar with the polarized. But when we take sections from the cervical root, the cells become cuboidal, and in the apical root or mid-root to apical root, they become spindle-shaped. What are the functions of the odontoblasts? Dentine secretion, of course. They work as a barrier, reducing toxins to reach the pulp. They allow fluid from the pulp to enter the dentinal tubules. Remember the extracellular fluid we talked about last week? They control the composition of this fluid. Also, they produce pro-inflammatory mediators and cytokines in case of injury or stimulus. As I have mentioned, there's different kind of cell-to-cell junction between the odontoblasts and the odontoblast and fibroblasts. These are the cell junctions, desmosomes, tight junctions, and gap junctions. You have discussed these in details in the cytology subject last year, so the details are mandatory to read from the book. The odontoblasts continue to lay down secondary dentine in a slow rate throughout life. This is an information we talked about last week. That's why the pulp chamber and the root canals become smaller and narrower with age. The odontoblast layer becomes flatter with time, So they are columnar at the beginning, then become cuboidal, and then spindle-shaped with aging, not only by the location, as we discussed earlier, and the number of odontoblasts also declines by apoptosis. The second kind of cells is the most ubiquitous cell, which is the fibroblasts. The fibroblasts, as you can see here in these slides, they form a loose connective or loose network throughout the tissue. They are linked to each other by adherent tight junctions and gap junctions. They have variable morphology. So, for example, here they are spindle shaped. Here they are star-like shaped. They, of course, connect to each other and to odontoblasts. What are the functions of the fibroblasts? So the fibroblasts actually produce fibers and ground substance for the connective tissue, and they degrade the matrix they formed, which means they make a turnover of the extracellular matrix. They also produce growth factors and cytokines. What about immune cells? As I have mentioned earlier, there's T cells, macrophages, dendritic cells, and neutrophils. You have learned all of these cells, shapes, and histology in the general histology subject. So we are going to mention them briefly. So the T lymphocyte, as you can see, they are found in small numbers in the normal pulp, but they increase if the pulp is injured. The macrophages, you know that there is different kinds of macrophages that they have different morphologies depending on the resting and non-resting form. They are widely distributed in the bulb, and they are denser around blood cells and odontoblasts. The dendritic antigen presenting cells, they are branching cells. As you can see, they are in close relationship with the odontoblast. They lie beneath them, between them, and they actually extend sometimes. They extend their processes inside the tubules, and sometimes when there is an insult or an injury, they actually extend their processes deep inside the tubule, as you can see in this histological section. This is a special stain to show you this beautiful dendritic antigen-presenting cells. What does this cell do? They actually look for antigens. They catch the antigen and then they give it to the T cell, which actually do the rest of the immunological reaction to defend the tissue against this antigen. The last kind of cells that's inside the dental pulp is the stem cells. There's a lot of different populations in different locations in the dental pulp, not only mesenchymal stem cells, which are discussed in your book, but actually there is embryonic stem cells, there is neural crest stem cells, a lot of populations. These are found mostly in the subodontoblastic layer. The subodontoblastic layer contain a special kind, which is the odontoblast progenitors. A progenitor mean a cell that has the capability to differentiate to only one kind of cells. So these are progenitor, but they are not fully differentiated. When a proper stimulus is there, they transform or differentiate into odontoblasts. These stem cells could be isolated in the lab and directed to differentiate into different cell types for regenerative purposes. Now we are going to discuss the blood vessels or the blood supply of the dental pulp. As I have mentioned earlier, the blood supply and the nerve supply come together as a bundle, the neurovascular bundle that enters the tooth from the apical foramina in the root and branches in the periphery and in the crown. So there's arterioles, venules that branch the whole way until they reach the coronal part where they branch profusely and make a plexus. There is larger vessels that almost 150 micrometers in diameter. And when you look at histological section, you will know that this is actually big. There is terminals of sympathetic nerves in association with the arterioles because these sympathetic nerves control the arteriole vasoconstriction by controlling the smooth muscles of the arteriole. The pulp has a very high pulpal blood flow, which is indicated here as 20 to 60 millimeter per minute per 100 gram. That is very important to create the high fluid pressure that is pushed to the dentinal tubules as a defensive mechanism to push all the pathogens away from the dental pulp. The profuse branching that happens under the odontoblasts, which makes this beautiful plexus, is called subodontoblastic plexus. That actually is under the odontoblast, between the odontoblast, and even between the odontoblast and the periodontium. The capillaries, some of these capillaries are fenestrated and they have only basement membrane above them, which makes perfusion easier. This is another histological section with a special dye to show you how the trunk enters the tooth as a big trunk here, and then the branches become smaller and smaller, then they turn into capillary plexus that actually become so profuse under the odontoblastic layer. In a cross section, you are going to see the blood vessels as circles, like this, with the lumen, maybe empty. Sometimes you see blood vessels. If it is an arteriole, you're going to see a thick wall because of the muscle around the vessel. If it's not, you're going to see a thin one. We will see it in a minute in other sections because the venule has a thin wall that doesn't have any muscles around. In a longitudinal section, this is how I are going to see capillaries. You'll get familiarized with the shape as long as we discuss these later, and actually, moreover, when you learn the practical module. So this is a really high magnification of an arteriole showing us the endothelial cell that forms the lumen and the muscle cells around it, which makes the arteriole look really thick in histological sections. Of course, as any other tissue, there is some lymphatic tissue inside the dental pulp, but these are really small lymphatic vessels, and they are not numerous. They are exist, but they are hard to observe. It's not really easy, for example, to say that this is a lymphatic vessel. Sometimes we will think this is a venule because the venule has the same shape. Maybe sometimes if there is a different lumen than RBCs, we can say, oh, okay, these are not RBCs, so this is a lymphatic tissue. This is here, as you can see, is the lymphatic tissue. It's really hard for you to notice or identify. But later on, when we learn how to identify this structure, which is the nerve, and this structure, which is the blood vessel, we will start, or you will start actually a little bit to learn how to differentiate them from the lymphatic vessel. Anyway, these are the lymphatic vessel, as I have said, are really hard to identify. So most probably I'm not going to ask about them in the exam, but the nerve tissue and the blood vessels, arterioles and venules are mandatory to know very well. The nerve supply of the dental pulp. So the nerve supply into the pulp, as usual, from the apical foramina with the neurovascular bundle. The dental pulp is heavily innervated, and just like the blood supply, it branches and branches at the periphery of the root and in the crown. They branch coronally, profusely to make a plexus called plexus of Rashko that is evident after eruption. As you can see, this is a special stain. This is the nerve trunk, showing us the subodontoblastic Rashko plexus, as you can see here. This is a trunk here and there, and this is the plexus. These nerve terminals here, they become so small and they come and enter inside the dentinal tubules. These nerves are non-myelinated, which makes the catching of the stimulus easier for these nerve terminals. Remember this section from last lecture? This is the electron microscope of the dentinal tubules, where we can see the dentinal tubule with the odontoblastic process inside and some axons, terminal axons inside the tubule. Here also, if you don't see it very well, there is a nerve terminal coming inside the odontoblastic layer. I always put the pictures with their captions, so you can go back to the original picture and take a look at it. Remember that the nerve fibers inside the dental pulp has actually two types. 25% of them are myelinated afferent, which are responsible for the sharp pain, and 75 of them are unmyelinated C fibers that are the majority of the nerve fibers and responsible for the dull pain. But don't forget that there is a little bit sympathetic efferent nerves responsible for the arterial smooth muscle contraction. Let's come now to the different regions of the dental pulp. So if we take a section of the dental pulp, we will notice that there is 5 different regions to study: the supraodontoblastic region, the odontoblastic region, the subodontoblastic region, which is composed of cell-free zone of Weil and cell-rich zone, and the bulk of the pulp. As you can see here, this is the layer of the odontoblast. This is small area above it, marked by the black arrows, are the supraodontoblastic layer. Supra means above, so the name explains itself. Underneath it here, this is the cell-free zone. We will discuss them in details in a minute. So as you can see here, this is a really high magnification, shows the odontoblastic layer, this area of supraodontoblastic layer between the odontoblast and the predentine. They appear because sometimes when we prepare the section, a little bit shrinkage happens. So in this area, what we find unsheeted axons, the one that I showed you just a minute ago, and we find dendritic antigens presenting cells, processes, or cells sometimes. And these are the regions. I like this photo, so I said to myself, maybe we have to show it to you. Then under the odontoblasts, we discussed the odontoblastic layer. Under the odontoblast, there is what is called cell-free zone of Weil or Weil. Actually, the proper name to be is the anuclear zone because there is cell bodies here, the processes of fibroblast, the odontoblasts, the axons and capillaries crossing this region, but the stains doesn't expose them. So it's not empty, and most probably the cause of it is the shrinkage that happens because of the processing of the tissue. We don't see this layer in the radicular pulp. Underneath it is the cell-rich zone. You find this area is really full of cells. It's either because the shrinkage that happens pulled the cells from the free zone to the rich zone, so making it rich, or because the capillary plexus, the nerve plexus, and a lot of other cells are in these areas, making them rich in cells. And there is the bulk of the pulp. As you can see, the bulk of the pulp is formed by the extracellular matrix with the fibroblasts. And there where we find the capillaries, arterioles, venules, nerve supply, and lymphatic supply. We will see a lot of sections for the bulk of the pulp in the practical module. Just another section to just remind you with the different layers. So we have the odontoblastic layer, the supraodontoblastic layer, the cell-free zone, the cell-rich zone, and the bulk of the pulp. And as you can see, there's blood vessels here and there. And we will see a lot of histological sections talking about the regions of the dental pulp in the practical session. What happens to the pulp with aging? As you can see in this picture, as the pulp age, the secondary dentine occupies the crown and root chamber, and the pulp becomes, or the pulp chamber becomes smaller and narrower in the crown and the root. Of course, as any other aging process, the nerve supply decreases, the blood supply decreases, the tissue become more fibrous. And in the dental pulp, special thing happen, which are calcifications. Calcifications happens as pulp stone or as snowstorm calcification that we will see in a minute. So as you can see here, more fibers, more collagen fibers, big ones are secreted into the dental pulp, which means less cellularity. The cell number become less. See the difference between the young pulp and the old pulp. Less cells, more fibers, less innervation, and less blood supply. Also, calcifications happen. This picture or this image shows us irregular calcifications here and there. That is called snowstorm because it looks like snow fricks here and there. The other kind of calcification in the pulp stones, as you can see in this picture, they can come in single or in groups. They can be detected on radiographs, as we will see next. You can see them on radiographs. They can be true denticles, which resemble dentine, like here. There's dentinal tubules inside, or they can be false denticles that look like bone. They have cells inside and they are lamellated. They have circles inside of them. They could be large or small, and they can be attached to dentine, as we can see here, or free, not attached to anything. And they can be actually embedded inside the cementum also, okay, inside the dentine. Sorry, they can be embedded inside the dentine. The only problem of these is when the tooth needs root therapy, they make the root therapy complicated. As you can see here, this is a dental pulp stone. This is an embedded pulp stone inside the dentine. This is an attached one, is attaching to dentine, and this is a free one. Another histological section to show them. So this is a free pulp stone, an attached pulp stone. Again here. So this is a pulp stone, a false one, because it looks more like a bone than dentine. Here too, false ones. There's cells and they are lamellated. As you can see in this picture, there is multiple ones in this tooth. The last kind of calcification is this snowflake, which means random calcification throughout the pulp, as you can see here and here. So this is all for today. This is the end of our lecture, and now I am going to be open for your questions.